In this video, we are going to look at the advantages and disadvantages of light versus electron microscopes. So, by the end of this session, you will be able to describe the advantages and disadvantages of using a light microscope and electron microscopes. You will be able to explain why light and electron microscopes have different advantages and disadvantages, and you will be able to justify the use of light and electron microscopes for different objects. So first of all, I would like you to recap some microscope definitions. We'll be using all these words in the PowerPoint today, so it's important you understand them. The words I would like you to look at are image, object, resolution, and magnification. Have a go at writing out the definitions, pause the video, and then you can check your answers. So here are the answers. You have the image, which is the appearance of an object or material when viewed under a microscope. You have the object, which is the material that is put under a microscope. Resolution is the minimum distance apart that two objects can be in order for them to appear as separate items. And magnification is how many times bigger the image is when compared to the object. And here is the equation for magnification is image size over actual size. There is another PowerPoint on magnification equations, so do make sure you look at this if you do not understand the equation. So, another little recap is how does a light microscope work? So you can see at the bottom of a light microscope, there is a light source. Above that you have a condenser, which controls how much light then passes through the specimen. The specimen is on a slide, which is held by the slide holder on the stage. The light passes through that specimen into your objective lens and then through your eyepiece lens, which then goes to your eyes. As the light microscope uses visible light, you can see your specimen in colour and it will not harm a live organism, so you can view live organisms under a light microscope. However, light microscopes have limitations. So the advantages are they have small and portable, you can see in colour, it doesn't kill live organisms so you can see them, and they are cheap to buy. Whereas the disadvantages is it only has a relatively small magnification of times 1500 and a poor resolution. And you can see the differences between cells viewed by the light microscope, with the ones on the left being cheek cells. You can see them in colour, you can see the nucleus and the cell membrane, but you can't see many other structures. Whereas on the right, you have a cell that's been viewed under transmission electron microscope. You can see the structures of the nucleus, the different organelles, including things like mitochondria and the endoplasmic reticulum. So what might you use a light microscope for? I want you to look at the different objects below and identify which ones you think you would use a light microscope for and why. So, looking at a root tip squash in college, using staining to study the structure of the alveoli, looking at the ultrastructure of a mitochondria, studying how tapeworms attach to the intestinal wall, and using Daphnia to see the effect of caffeine on heart rate. Pause the video now, and I would like you to write out why you think you would use the light microscope for which ones and why. Okay, which ones would you use? So looking at a root tip squash in college, I would use a light microscope for, so it's cheap and portable, so we can use them in college, and you are using stains to be able, so you need to be able to see in colour. If you can't see in colour, you can't see those chromosomes. And you have sufficient magnification here to study the chromosomes in a field of view of cells. So it is good to use a light microscope for that. Using staining to study the structure of the alveoli. When you're using stains, you need to see in colour. Also, to use a light microscope, it's relatively easy to prepare. So lab samples can be done quickly, easily, and relatively cheaply, which is really good in a medical lab environment. And then if you're going to look at the effect of caffeine on Daphnia's heart rate, you need to be able to keep that Daphnia alive. So you can observe live organisms, so you can look at their heart rate. So we've talked about our limitations, these being the maximum magnification and the poor resolution. However, you can overcome these using an electron microscope, which has a maximum magnification, theoretically, of times a million, and a maximum resolution of 0.1 nanometers. However, in reality, due to technical difficulties, So things like you can damage the specimen with too high an electron beam. It's not always possible to reach maximum magnification and resolution. Disadvantages of electron microscopes: they're not portable, they're expensive, they only show black and white images, you cannot view live specimens, there's a very difficult preparation process for your specimen, and they produce artifacts. Artifacts are caused in the preparation process, and they can appear to be part of a cell when actually they are not, and they cause problems in analysis of data. So, instead of using light to show their images, electron microscopes use electrons, and this is why they have a much higher resolution. So let's recap how transmission and scanning electron microscopes work. So here is my transmission electron microscope. So at the top, you have a high voltage electron gun, which produces a beam of electrons. The beam is focused on the specimen by a condenser electron magnet, which are the condenser lenses in the picture. In a transmission electron microscope, the beam passes through the specimen, so the specimen must be very thin. Dense parts of the specimen absorb electrons, so they don't allow the electrons through, and these appear dark. Less dense part of the specimen allow electrons to pass straight through. The electrons that pass straight through hit the fluorescent screen and bleach it. And then you can take the screen and photograph it to produce a photomicrograph. You also have a scanning electron microscope. So scanning electron microscope, rather than looking, say, inside a cell, will look at the surface of an object. So in a scanning electron microscope, the electron gun produces a beam of electrons. This beam is again focused by condenser and objective electromagnets, which are called lenses in the picture. The beam is passed back and forth across a portion of a specimen in a regular pattern, and then these electrons are scattered by the specimen, and the pattern of this scattering depends on the contours of the specimen's surface, so it creates a 3D picture of the specimen surface. You build this 3D image by computer analysis of a pattern of scattered electrons and your secondary electrons produced. And secondary electrons are electrons that are from atoms inside the sample, and these have jumped out of the sample because they have been excited and ionized, and they are also detected. So why do electron microscopes have a much higher resolution? We've talked about the fact that they use electrons, but why does this actually provide them with a higher resolution? Well, it's to do with wavelength. So you can see in the diagram, the wavelength of visible light is 750 nanometers to the red end to 390 nanometers at the violet end. However, your 60 kilovolt electrons have a wavelength of 0.003 nanometers. So therefore, you are going to get a much shorter wavelength. The shorter that wavelength is, the shorter the distance you can determine between 2 points, as shown in this diagram. So if you have a long wavelength, you can't determine between 2 points that are very close together, so you have a poor resolution. Whereas if you have a short wavelength, you can determine between 2 points that are much closer together, so you have a good resolution. With a little bit of physics in there, if you have a long wavelength, you have a low frequency because you have less waves in a short time. However, if you have a high frequency, you have more waves in a short time. So the shorter the wavelength, the higher the frequency. So why might you use an electron microscope? Again, look at these pictures. They're the same as the ones you've seen before. So that's your root tip squash in college, your staining to study structure of alveoli, the ultrastructure of mitochondria, how tapeworms attach to intestinal wall, and using Daphne to see the effect of caffeine on heart rate. Look at these images and think which ones you would use an electron microscope for and why. So pause the video, write it down, and have a think about which ones you'd use. Okay, so you'd use it for looking at the ultrastructure of a mitochondria. So look at the tiny structures that make up organelles. You would need to use a transmission electron microscope. You need to be able to see inside that organelle, and to do this you need high magnification and resolution. To study how tapeworms attach to the intestinal wall, you want to look at the surface of a tapeworm. You want to look at the structures that hook onto the intestine, so you need a scanning electron microscope. Both because it's got a high magnification and resolution, but also you want a 3D image of the surface of the tapeworm to look at how it attaches to the intestinal wall. So you should now be able to describe the advantages and disadvantages of using a light and electron microscope, explain why light and electron microscopes have different advantages and disadvantages, and you can now justify the use of light and electron microscopes for different objects.